Hello. I'm doing the exact same experiment as Aditya, except I'll be telling you a little bit more about what's happening when you're adding all the ingredients we're adding. And I'll also be using a banana instead of an orange. So the first step, same as Aditya's, is to cut up the banana. Or in Aditya's case, orange. So I'm just going to cut this up. It just needs to be enough so that I can put it in the blender. You want small pieces because a banana is a bit more chunkier and you want to get it as small as possible. As Aditya said, you want a juice. You want a soup-like material instead of just an ordinary chunky smoothie. The reason behind this is because we want to isolate all the DNA. If we have a really chunky surface like this, we can't really do that. But if you have something smooth, then it's a little bit easier to do it. Also, by doing this, we're separating the different parts of the banana. So, like Aditya's experiment, put all the banana pieces, these slimy banana pieces, inside. Also, we're adding 2 tablespoons of water. 1, 2. I'm sure all of you can count. I'm just going to wipe my hands a bit. Because now we're going to add the salt. Should have done this before. Okay, some salt now. 1 large pinch of salt. And I might just add a little bit. There. So now, after the salt, we're going to blend it. Now, if you need help with this, sure, go ahead. Ask your parents. But it's fine to do this step alone. When you get to later parts, you might need some parent assistance. So, lock this in tight. You want your DNA samples to be intact. And blend it for about 15 seconds, like Aditya said. You want it to get it. You want a smooth texture instead of a chunky texture for this. Now, for this part, we need to strain out the cell membrane, as Aditya was saying. It's the same concept of isolating where the DNA is. Because when you're doing this, you're taking care of some of the lipids in the cell membrane and pretty much taking out the chunky part of the cell membrane. This is a bit, like Aditya said, some of the more gross parts of this, because you actually have to use your hands to get all the juice out of the banana. And especially because the banana is so chunky, it's going to be a little bit harder. So, now after you get the banana juice, what you do? Add the soap. So you want this. So now the soap. The soap is what gets rid. The soap just breaks down the cell membrane even more. Because the cell membrane is extremely minuscule. And even though straining took out most of it, we still want to get out all of it. We want to make sure we get out all of it. So now, we're going to wait 10 minutes. And wait for this to get ready. Okay, the time is done. Now, we move on to the next step. Now we've got to add the enzymes. The pineapple juice. As before, Aditya used. But this time, we're going to use it for bananas, obviously. We are using pineapple juice, by the way. It's not any other type of solution. So we pour it in. Mix it up a little bit. Shake it down, mix it up. Do the little thing. So we do this, right? It's mixing, keep on mixing. So after, we just let it just mix, keep on mixing for about a minute, right? This should be sufficient enough. Now what we do, pour the mixture. The enzymes, by the way, what they do, they get rid of all the proteins. They don't really get rid of them, they cut them up. Because at the center of the cell is the nuclear membrane, and that's where all the DNA is stored. So, now we're going to pour it into this medicine bottle. Just not all of it is necessary. And I'll probably be klutz and pour half of it down on the ground. So we don't need all of this. We can pour a little bit out. Now for the alcohol. Remember, this is a very complicated step, though it may not seem so. Alcohol is pretty dangerous. So, and this is 91% alcohol, so it's pretty big. And all of those people who just can't wait till they're 21, try to restrain yourself here. So we're gonna put about the amount that's already in here of alcohol into this. Okay, so we put, as Aditya said, 5 milliliters. So there, poured it in. It's good. So now, if you can see really closely, the DNA is suspended, while the banana juice is down here, and the DNA is suspended in the alcohol. You might not be able to smell this, but this smells really bad. So now, here comes in the toothpick. We're going to use the toothpick to get all the DNA out. So, you want to do a twisting motion, along with something close to pulling up. I can't get all of the DNA because there are a lot of it in here, in this solution, but I'll show you a little bit of it. Like Aditya described it, it's pretty much snot. It looks like snot. You see here? This part is where the DNA is suspended inside the alcohol. Compared to strawberry, this has a lot more DNA. For example, I'll show you just a small sample of where all of the DNA here. Here's just a small sample of it. As this is described, it's a snottish-like substance, and all of the white stuff suspended in the alcohol is also DNA. And now that's the end of the experiment. You can just do whatever you want it with the DNA. This is our conclusion. As you can see, there was clearly more DNA in the banana. My conclusions, along with Aditya's, are that even though we used the exact same procedure, we got different results. The only variable in this experiment was the types of fruits. And we can clearly see the banana yields more DNA. All of this in the clear liquid, suspended in the clear liquid, is DNA. We come to the conclusion that the banana holds more DNA. And this may be due to the fact that it's more condensed versus the orange, which is less condensed. Thank you for watching our amazing scientific discovering cook show.